to welcome everyone to this session on recent advances in electrodiagnosis and therapeutic pipeline in periodic paralysis. We all appreciate you staying to the end and coming here to an 8 o'clock session on Saturday morning. And I'd also like to thank, at the outset, the Periodic Paralysis Association for sponsoring us to come and share this information with you. So there'll be two presentations this morning. We're going to start with a talk by Karen Suetterlin, who's a lecturer in clinical neurophysiology and a member of the AGE research group in the John Walton Center for Muscular Dystrophy at Newcastle University. And she'll be speaking on muscle velocity recovery cycles in periodic paralysis, deep phenotyping tool, and potential biomarker from bench to bedside. Karen, it's all yours.
So, firstly, thank you for everybody for coming and to the Periodic Paralysis Association for the talks today, and for Dr. Cannon for the invitation. So I have nothing to disclose for financial disclosures, and I think, as this audience will probably know, the skeletal muscle channelopathies really form a spectrum of disorders, so ranging from those that predominantly manifest with myotonia, the non-dystrophic myotonias, seen over on the left-hand side, your right-hand side of the screen, and those that manifest predominantly with weakness, the periodic paralysis on the right. And underneath, you can see, according to that arrow, the membrane change that results in these phenotypes, so one of membrane hyperexcitability in myotonia, or membrane inexcitability with the periodic paralyses. And really, genetic diagnosis is the mainstay, so the first step in diagnosis of these patients. And just to mention, within the periodic paralysis phenotype, there's also Anderson-Towle syndrome, which, in addition to the prominent weakness, also manifests with extra muscular features of the cardiac arrhythmias, so particularly long QT syndrome. And there's this triad of dysmorphic features, cardiac arrhythmias, and periodic paralysis, and the three can be variably penetrant. So the dysmorphic features can be extremely subtle, sometimes only obvious in retrospect after you've made the diagnosis. And so in any patient really presenting with a periodic paralysis-like phenotype, we would suggest they always have an ECG to look for that. But with the advances of genetic diagnosis, as you'll know, new genes have been identified with a periodic paralysis-like phenotype, and we were discussing some of these yesterday. One of those is the ranitidine receptor, and now that is a huge gene that's also associated with large amounts of normal variation and a number of different phenotypes. In those patients where it was associated with periodic paralysis, the majority of those also had other features. And similarly, the T-tubule sodium-potassium ATPase, has been a single case report there of a patient with that, but again, also had CNS features. But as well as new phenotypes new genes associated with periodic paralysis, we also now have new phenotypes associated with the known gene. So the sodium and the calcium channel can also cause a congenital myopathy, and also the previously described congenital myasthenia. So what I just want to highlight really is that the muscle sodium channel mutations in that can cause any one of these phenotypes, ranging from pure myotonia to the myotonia weakness overlap to pure weakness, and also the congenital myopathy and congenital myasthenia. And whilst the improvements in genetic testing over the last decade, sort of going from doing point mutations as the routine to what were hotspots, and then whole gene, gene panel, exome, and genome, that's led to incredible advances in our ability to diagnose these patients. It's also led to greater complexity. So if you imagine when you're looking at a point mutation, maybe have one or two variants, increases with hotspots, whole gene, panels, exome, up from about 1 or 2 with a hotspot up to about 4 million variants with a genome. And so we're faced really with this avalanche of genetic data. And the majority of those variants will be easy to filter out or exclude according to where they are. But there will be still a number of variants that may or may not be clinically significant, and it's difficult to determine. And so I think we really need to develop better tools to try and understand which of these variants are contributing and which not. And in the case of the channelopathies, a functional marker, and ideally one that can be applied in vivo, so you can understand the effect of a specific mutation within the context of a specific genetic and environmental background of the patient in front of you. So historically, for the channelopathies and for periodic paralysis, the functional test has been the long exercise test, and we heard a little bit about this yesterday. This is a long exercise test shown for a patient with hypo-PP. The filled circles represent the CMAP area, the empty circles represent the CMAP amplitude, and you can see, so there's this period of baseline recording, and then you'll exercise. We tended to do the adductor digiti minimi, so for 5 minutes. I've done this, it's really hard to do. It's very tiring. You have to do 5 minutes. We tend to do 15, 20 seconds, followed by 2 seconds break. The difficulty in that is as people get weaker, they tend to move their whole arm, and your stimulating or recording electrodes may move, which will give you a sort of artificial drop. But what the main parameter we're looking for is a 40% decrement from the peak CMAP amplitude. And so the considerations with that, as I said, is the moving electrical, the electrodes, but also if you're doing a short exercise test as well to make sure, as was mentioned yesterday, that you do it in the other hand, otherwise you may miss a significant decrement. But this is some work that we did on the cohort in England, in Queen Square, when I was there during my PhD. And what you can see on the y-axis is the percentage, and on the x-axis here, the patients with genetically confirmed periodic paralysis. So all of these patients have genetically confirmed periodic paralysis, are grouped according to their frequency of attack. So on the end there, it's weekly or daily, and all of those patients had a positive long exercise test. This was from monthly up to a couple of times a year, and about 70% of those. So these are 30% genetically confirmed periodic paralysis with a negative, and here it's around 50% with patients who have infrequent or haven't had an attack. And that has been also shown in a separate study from an independent group. So again, here, patients who had a recent attack have a positive long exercise test, but those who haven't had a recent attack, it looks similar to controls. Similar thing for the thyrotoxic PPs, so here pre-treatment, there post-treatment. And also what I want to highlight here is the so-called carriers, the asymptomatic carriers of PP, again look similar to controls. So the long exercise test has been very useful. It obviously is a functional measure, but it relies on inducing a localized attack of paralysis, so it's an ictal measure of the membrane function. Sometimes when you're doing that, because of the long rest afterwards, there are occasions where it can then go on to a generalized attack. And it also requires or relies on the voluntary effort of the patient. So I think we ideally want an interictal measure of membrane function that doesn't rely on the voluntary activation of the patient. And about 40 years ago, actually, there's a group in the Netherlands who did this study on a large family of hypo-PP, and they used muscle fiber conduction velocity. And what they found is that they were able to completely distinguish the controls from the patient, but also, shown in this slide, actually, if you look at the filled circles are controls, the empty circles are hypo-PP, and the crosses, which you may not be able to see that well, are the so-called asymptomatic carriers. And there were a number of asymptomatic carriers that clustered down here with the PPs. 2 of those 4 had biopsies, both had vacuolar myopathy. And the other ones either went on to have attacks or to develop the myopathy associated with periodic paralysis. So this detects interictal changes that aren't apparent on the long exercise test, and importantly, there are people with periodic paralysis and gene effects who don't necessarily have the attacks of paralysis. But I think now, with genetic testing, we're not really looking to distinguish the patients from controls. We really want to be able to distinguish different types of channelopathy and different biophysical changes in the channel. And whilst this looks promising, the PMC patients that were studied also had a decrease in conduction velocity, so weren't distinguished. So the next step really that you can do is look at what's called the velocity recovery cycle. And so it's been shown that conduction velocity in skeletal muscle closely parallels the excitability, and that's based on this phenomenon of an after depolarization. So if you have a skeletal muscle action potential that you can see here, After, it doesn't go back to baseline, you have this little hump called an after depolarization. If you trigger a second action potential at any time from about 6 milliseconds to up to 1 second after that, it will conduct faster than the first action potential. And that effect will summate, so as the number of action potentials increases, the amplitude of the depolarizing after potential also increases, and eventually you'll get some membrane depolarization. And I think the way of thinking of this in general for the velocity recovery cycles is that conduction velocity is affected by preceding events. So linking that to my own life, a question that I'm commonly asked by my children, can we have an ice cream, is this has been the preceding event, and the answer is no. Whereas on the rare occasions where I've seen something like this, it might alter the outcome. And it's essentially the same thing. So we are looking at post-impulse change. There's no ice cream involved, but we are looking at post-impulse changes in conduction velocity. An increase in conduction velocity we see as a reduction in the latency from the test stimulus to the peak of the response, and that's known as supernormality. And that the extent of that effect will vary according to the interval between a conditioning and a test stimulus, and also according to the number of conditioning stimuli given. And we plot that out according to the interval between the conditioning and the test stimulus to get the recovery cycle. And these were done by Stahlberg initially and then by Mihalin in 91, but they did it as single fiber. So they had 2 electrodes within the same fiber and then looking at changes in velocity recovery cycle. I think in this paper they looked at 8 volunteers, they studied 70 fibers, from which they got 16 recordings that were reusable, and these were very variable. So as they said there, it was technically extremely challenging. So in around 2009, Hugh Bostock with Werner Zigragan, I think inspired partly by the C-fiber velocity recovery cycles, adapted this. So they've made it a multi-fiber velocity recovery cycle. They're known as MVRCs. The M stands for muscle, but I think you can think of it as multi-fiber as well. This is a picture of the setup. So, you have a direct muscle stimulation with a monopolar electrode and a recording concentric electrode. There's a link here to a Jove article which describes how to do it. This is using the Q-Track software developed by Hugh Bostock, but it could also be set up with stimulation protocols, I believe, on standard kit. And this is really just to say that these are submaximal stimuli that you're using. And the fact they're submaximal enables you to look at more conditioning stimuli because you're not then evoking a big twitch. It also makes it more comfortable. And the reason it can do that is because unlike the peak, which will gradually increase as you increase the stimulus amplitude, the latency remains pretty stable. And it's the latency that we're looking at with the MVRCs. So they've been done in brachioradialis, they've been done in tibialis anterior, they've been done in rectus femoris, there's an N of 1 for orbicularis oculi, and there's also frontalis. So it's just to say they can be done in many different muscles, and they are very well tolerated. So just to show you a kind of get to some of the data, but first just to show you the graphs. So they're plotted on the x-axis, you've got the interval between the conditioning and the test stimuli, and on the y-axis here is the latency. So a reduction in latency means it's going faster. We've referred to that as supernormality. An increase in latency isn't really seen in human MVRCs, but that's referred to as subnormality. And this is a recording from my TA. And you can see in red, that's the response to 1 conditioning stimulus, in green to 2, and in blue to 5. And what hopefully you can appreciate is that there are different phases to it. And the 2 phases are differentially affected by increasing the number of conditioning stimuli. And so the parameters that we look at related to that is the point at which the latency of a conditioned and an unconditioned stimuli are equal. And that's because it reflects the end of the relative refractory period. So if you imagine prior to that, there's a period where there's no response, so that's the absolute refractory period. In this period, it's slower, and then at the point where that slowness ends and it starts to increase, that's the relative refractory period. This early phase of increased conduction velocity is known as supernormality, and the later phase is known as late supernormality and believed to reflect more the activity-dependent T-tubule accumulation. And then at the end, there's residual supernormality. So how does this relate to the periodic paralyses and the channelopathy? So I'm going to talk about work. This was done by Veronica Tan, and looking at patients in the UK. We've got a national skeletal muscle channelopathy service that's led by Professor Mike Hanna. So she did a study that looked at 7 patients with hyper-PP and 10 with hypo-PP, of which 2 had the sodium channel variant. And what I just want to point out is that of these patients, several of them, which you can see in this graph, I think 3 of the hyper-PP and at least 1 of the hypo-PP had a negative long exercise test. So this is what their MVRCs look like. This is a graph looking at 1 conditioning stimulus, and this is at 5. And if I just focus on the 1 conditioning stimulus first, I just want to highlight this region where you can see that the patients shown in blue with hypo-PP, sorry, on the wrong thing, have a longer relative refractory period, whereas the hyper-PP is more similar to the control. And that really is consistent with a depolarized resting membrane potential. In keeping with that, they also have this reduced supernormality here. And then you look at the response to 5 conditioning stimuli, that becomes more evident, especially in the late supernormality. The hyper-PP patients have an increased residual supernormality. That's the amount of supernormality left after 1 second. And also, if you look at the area under the curve, the total is increased, which is kind of consistent with the described window current, which if you have any questions about, I'm sure Dr. Cannon would be very happy to answer. And this is just to show for Anderson-Towle syndrome. It looks similar to that we see in hypo-PP, which I think is as you'd expect from phenotype. But the relative refractory period is slightly longer and the supernormality reduced. This is a slightly different parameter which gives additional information. And previously I said as you increase the number of stimuli, the depolarizing after potential summates, and you eventually get some depolarization of the resting membrane potential. So this is using a frequency ramp. You have a period of baseline stimulation at 0.5 hertz, and then you increase over approximately a minute up to 30 hertz, and then look at the recovery. And what you get in the response for latency is you get a U-shaped curve, but in the hypo-PP, that increase in latency is reduced. In the hyper-PP, it's similar to control, so it wasn't significantly different, but there's a trend that it's increased. And so when you combine these parameters, you can't completely distinguish either hypo or hyper-PP from controls, but you can distinguish them from each other by combining the response on the frequency ramp and some of the MVRC parameters. And so I think that's useful in terms of thinking of the phenotype, as I said, the sodium channel being responsible for any one of the phenotypes in muscle channelopathies, so helping to distinguish people with a sodium channel mutation what their phenotype is likely to be. And just with that, just to show briefly some of the recordings that Veronica did in the sodium channel myotonia and paramyotonia patients. So just what I want to highlight on the sodium channel myotonia is that their MRRP is actually reduced compared to controls because they recover from inactivation quicker, so they could be differentiated from controls because of a reduced MRRP. Whereas on the bottom of that graph here, the PMC ones perhaps look slightly similar to hyper-PP. And as was mentioned in the session yesterday, the kind of clinical overlap between PMC and hyper-PP is quite great, and it's very difficult to distinguish them clinically. But actually, using the frequency ramp in combination with the MVRC, these were patients with mutations in T13, the classic PMC mutation, T1313M, they could be completely separated, so you could completely distinguish them from hyper-PP. Now, obviously, this is in a single cohort, and this is in our cohort, and patterns were previously described with the long exercise test that weren't replicated elsewhere, so we do need to repeat it in other cohorts, but it at least looks promising in terms of distinguishing sodium channel phenotype. So I mentioned it might be a useful measure also for looking at variants of uncertain significance. So sometimes in patients where the phenotype doesn't quite link to the genotype or the known genotype. And this was a patient at the Queen Square Channelopathy that we saw there. And he was a Kurdish refugee who, at the age of 17, he presented, he had really quite pronounced and severe myotonia, really affecting his hands and legs. And on examination, he had some hand grip myotonia and a bit of eyelid myotonia, but was really quite disabled by this. He was started on mexilatine, which he had a little bit of benefit from, but despite that had severe myotonia ongoing. And about the age of 25, he started to develop some episodes of weakness. So short duration, about 5 to 10 minutes, mainly in his legs and triggered by long rest after being seated. He was under investigation for quite a long time because he didn't have, at that time, any mutations in the classic spots. And then when we got to whole gene sequencing, sorry, this is just to show that he had myotonia, he was found to have a mutation in the sodium channel S4 domain, arginine of domain 1. And these are mutations that are classically associated with hypokalemic periodic paralysis. And in fact, the same residue but mutations to different amino acids at this point were associated with hypo-PP. But his phenotype was of really quite severe myotonia. So we did expression studies and we looked at that variant and we found that it had enhanced activation, which would help explain the myotonia. And there was a gating pore current as well, which you see with hypokalemic periodic paralysis, but it wasn't as severe as the other mutations at that. But it still didn't explain the severity of his myotonia. So this gentleman on his panel also had this synonymous variant identified in his chloride channel. And when we first identified it, we thought it was unlikely to be significant, but then later found several patients with recessive myotonia congenita who had this variant. So we did the MVRCs in this gentleman to try and see whether or not that chloride channel variant might be having an impact and trying to explain the severity of his myotonia. And so what you can see, his MVRC is shown there in red, and hopefully you can appreciate that the extent of supernormality is greater than the other MVRC patterns which are shown there for the groups of patients for PMC, sodium channel myotonia, and myotonia congenita. And he also had much more, it's difficult to appreciate there, but increased residual supernormality compared to the other sodium channel myotonias. We did a frequency ramp on him, and on that he was in his own category. He's kind of different from all of them. And then you can do groupings based on the parameters of the different testings. And what was interesting from that is he tended to cluster either in the myotonia congenita group for these, or on his own as an outlier, but not with the sodium channel caused myotonia. So we felt that this provided pretty good evidence that his chloride channel variant was significant, and that was really helpful in terms of giving him genetic counselling for the likelihood that his children would have myotonia as severe as his was. So that's sort of looking at these tools as a way of deep phenotyping. But what about possibly using them as a biomarker? So there's a group in Denmark who've done this looking at mexilatine. So this is using healthy controls and doing MVRCs pre and then 3 and 5 hours post mexilatine. And what they've shown is that it's sensitive enough, it can detect the effect, and the effects are consistent with the known activity-dependent block of mexilatine. There's also a study that was done by Werner Zagragan, and they did force training, so a 2-week force training, again in healthy controls, and they did MVRCs before the force training and 2 weeks after, and they showed small but very consistent and also significant effects, so in the reduction in the relative refractory period and an increase in early supernormality. So I think there's good potential there for MVRCs. So, I mean, it's appealing that they're point of care, they're relatively fast. I didn't say, but the MVRC and the frequency ramp takes 5 minutes to complete as a protocol, which is quite appealing when you compare it to a 50-minute long exercise test and also a 10-minute short exercise test with ice or longer. They're also relatively inexpensive. They're minimally invasive. I think it's a real bonus that they are interictal, so it doesn't rely on inducing an attack. they detect reversible functional deficit, and I think that's an important point. So MRI is a really important biomarker now in neuromuscular disease, but for the mainstay it relies on measuring muscle fat infiltration, which we believe is irreversible. And so I think that they are promising, and I think they are, promising in terms of distinguishing the different biophysical deficits. But we still don't really understand the MVR signal. So the parameters and the protocols we're using were developed based on the existing long and short exercise test and what we sort of thought might work. We don't know if we're measuring the best parameter, and we also don't know which ion channels and pumps exactly we definitively are contributing to which part of the MVRC. And so really, in order to get the best possible use from them, I think we need to explore that and understand that much better. And in order to do that, ideally, we'd have a good model that we could use to generate and test hypothesis and iteratively improve. So this is James Fraser from Cambridge, and this is work that he did that developing a model based on rat muscle, but of skeletal muscle excitability. And this is showing it sort of replicating some myotonia, and it was used in one of the studies Dr. Cannon referred to yesterday, looking at calcium and magnesium. But this doesn't predict supernormality. So there is a problem with the experiment, well, not a problem, but there is a gap in our knowledge and our understanding that we really need to address in order to, I think, take this forward. And so in order to do that, I think we need to go the wrong way, so to speak, down the translational pathway. And what we've done is reverse translate the MVRC so that you can apply them in animal models. And the big benefit of that is that you can combine it with a selective pharmacology, similar to the maxillotine, but obviously you have a wider range of tools. So this is published, but this was initial doing in mouse, TA, and combining it with blockade of the chloride channel in vivo. Now, the big advantage of this is that this is a channel blocker that has an established in vivo dose and doesn't have significant cardiac toxicity at that dose. But many of the blockers that we would like to use to understand this signal have cardiac effects, and those cardiac effects will cause ischemia, which will have its own problems with the MVRC. So really, in order to take that further, I think we need to now develop these tests ex vivo, so doing MVRCs on isolated muscle, and that's what I'm trying to develop now in Newcastle. So hopefully we'll have some data on that to follow soon. I think the other benefit of the reverse translation is that you can apply the same technique to your preclinical models and your human patients. And I mean, I think it's pretty obvious that there are going to be differences between mouse and human. I don't think that comes as a surprise to anyone. But then we can try and understand those differences in phenotype by comparing and understanding and exploring them, and possibly improve the translational output. However, there are several areas where mice recapitulate the phenotype very well, and I think Dr. Cannon will talk about the hypo-PP mice and some of the other ones later, but just to mention briefly some of the sex differences in phenotype of the channelopathies. So these have been described for a long time in humans, and it was quite striking that they're recapitulated in the mouse models to the extent where some of the down to mutations, so the mutations that have been associated with in humans are also associated in the mice. So this is a model of hyper-PP that isn't as common. So there's a couple of patients that we have with the mutation. It's an I582V. But the mouse model of it, what you can see is there's a clear sex difference in the penetrance of these paralytic episodes. So these are episodes of hind limb dragging that the mice have. So you can see that at 60 weeks, essentially all of the males have had an episode of hind limb dragging, whereas only just over half or under half, of the females have. And that is despite all of them. So there's full penetrance of myotonia on the EMG, so that can't explain it. There wasn't any clear cause identified in the initial study describing it. So what we wanted to do is to see whether maybe MVRCs might help us detect why that is. And the question really is this a reflection of a physiological sex difference in muscle excitability that makes perhaps the males more prone to the paralytic attacks, or is it something related to the mutation specifically? So this is looking at MVRCs from mouse TA, and in blue are the males and in red are the females. And what you can see is that in the males, there's this increase in supernormality in response to 5 conditioning stimuli compared to the females. This is 1 conditioning stimuli and this is 5. And what we found in the hyper-PP mice was that it looked like this effect was exacerbated. So again, the females are in red and the blues are in males, but it seemed like that increase in supernormality perhaps tipped them over, so they had a prolonged relative refractory period, suggesting there was a degree of depolarization, and they had reduced supernormality, both compared to the hyper-PP females, but also the wild-type males. And similarly, the hyper-PP females that had myotonia and EMG, but not the paralytic dragging episodes, so they had increased supernormality compared to the males here, but also the wild-type females, consistent, we think, with the myotonia-like phenotype. So, and these differences were muscle-specific and seemed to correlate with the muscles which had the hind limb. So there were hind limb dragging episodes. They were never observed in the forelimbs, and the sex differences weren't as apparent in the triceps. So really, I guess, what I would like to suggest is that we can benefit from using the same biomarker or the same measure of excitability across the translational spectrum. So, the patients are missing at the bottom there, so they've been cut off, apologies. But really, sort of developing and understanding the signal. So initially looking at in healthy controls, then looking at periodic paralysis patients, but now I think going back across the translational pathway to understand which ion channels are contributing and when, feeding that into a model with the idea that we can then start to design specific protocols and parameters to probe specific channels that we may be interested if there's a variant of uncertain significance, but also then starting to try to understand the differences in phenotype in terms of sex, also looking at age-related phenotype, and also using that understanding so that we are better able to apply and understand the signal for patients across their life course. So rather than just having a set normal value, being able to understand how you would expect it to change with age and sex. And the hope is that this may help us, firstly, to interpret some of the genetic data that we're getting, the variants of uncertain significance, possibly in the future, as the many therapies are coming, to use it as an outcome measure, or at least as a measure to validate that the target is engaged and that there's a sort of change in the functional effects. But also, as the therapies are coming, we want to be able to rapidly identify and diagnose patients to make sure that they're able to get the right therapies. And crucially, with these, we hope we may be able to identify which therapies they benefit most from. So with that, I'd like to thank many people that have helped with this work in Newcastle and in UCL and at home. So just to show you all the faces, so in yellow are the people in Newcastle, in white at UCL, and red at home. And yeah, thank you very much for your attention.
Thank you. That was great. I mean, it was fascinating and a bit complicated, so my question may actually be from a limited understanding. So there are 2 questions I have. One of them, so based on my understanding, this cannot be used to diagnose disease versus normal, right? Because that was not significant enough, but to categorize different disease types. Is that?
Yes. Yeah.
So if that is correct, how would you use this alongside the long exercise test or in place of it? And the problem would be then, how could you use this for a diagnosis or as a biomarker for entry into studies? The second question I had was about the mexiletine effect. Was it on normal muscle?
Yes.
So that would not reflect any clinical change then. It would just change. So how would you correlate that? Well, are there any studies done to see if this correlates with, or the degree of change to be used as a biomarker
So in response to the first question about how you could use it in terms of diagnosis, I think the first thing is we really need to replicate, I think, in an independent cohort, so to establish really that these changes are seen and are robust in different cohorts. I think personally that this provides extra information above and beyond the long exercise test, and I think when you think of the sort of the time involved and also the fact that you're not needing to induce an attack or relying on voluntary activation, my preference would be probably to do the MVRC in place of, but we don't yet have the data for that. So we're in collaboration with colleagues at Queen Square. So I'll show his picture. Oh, no. Anyway, there's the neurophysiologist Arjuna at Queen Square. We're going to start trying to do both at the same time to really try and compare them prospectively, and hopefully that will be able to address that question. But I think at the moment, as you said yesterday in the talk, really it's genetics first, and then where that doesn't necessarily answer the question, these tools are helpful, I think, to try and identify or help to see whether there's the deficit there to fit. With regards to the maxillatine, so Veronica did the study on the myotonia congenita patients, and she subdivided it into those. So there are study and there's data on those that were and weren't taking maxillatine, but it hasn't been done kind of prospectively and looking in patients who were how much benefit they got from maxillatine, and also timing it according to the timing of their dose of maxillatine. So I think again those would be useful studies to do.
One more follow-up question. So one thing what actually from your response came to mind is, can this be done on people who do? Can this be done on people who have a normal long exercise test and see if it adds additional value, even if you can't see a difference, any additional value to someone, for example, who has a normal test or non-significant test? That would also be interesting to me to know as an added value in those cases, at least.
Yeah, absolutely.
Thank you so much. Wonderful. Thanks.
Brilliant talk, really wonderful, great time management. We have time for questions. So I have a few. First, what would the learning curve be for an experienced clinical neurophysiologist to pick up this technique?
So I think in terms of actually doing the technique, it would be very quick. The software sometimes takes a little while to get used to, but an experienced neurophysiologist, I think, would be very able to have the setup and do that quickly. I think what takes time is getting used to the analysis, but again, I think that would be much faster in people that are used to it. So I think there's a group in in Denmark who apply MVRCs a lot to other conditions, so they're looking at critical care myopathy and other ones, and have done workshops where they've looked, I'm not sure for MVRCs actually, but for other ones, at how long it took and then how much inter-rater reliability there was.
Yeah. So that brings up another question. A potential wonderful advantage is to be able to use this in children, uncooperative patients, or even under light sedation or general anesthesia. Do you have experience for how sedation or anesthesia might impact this and change the reliability?
So obviously the mouse ones are done under anesthesia. I don't have a control under, didn't manage to do that. There's data, I think, for sort of some of the peripheral nerve measures or more central measures that it affects it. From what we could find, it looked convincing that it didn't have dramatic effects, but what I'd like to do is compare it in healthy volunteers sort of undergoing anesthesia for a different operation, or different, non-muscle condition, and to ideally see if you can do an MVRC under anesthesia just to make sure. But that's not been done.
I would just comment, from the fundamental underlying electrophysiology, there's a wonderful consistency here that the hypo-PP cases stand out as having features compatible with interictal mild depolarization, and that's a consistent story from many types of measures. So I think that speaks to the validity of this, which is wonderful. And then my last question, beautiful case example of this synonymous CLCN1 gene. So were you, because of the abnormal functional test results, motivated to go look at gene expression and mRNA levels of that and try to figure out whether total RNA was decreased in muscle from that individual?
I have to admit we haven't done that, but that would be, yeah, that would be a nice step. Thank you.
John Kincaid from Indiana. Great talk. It shows that classic electrophysiology is still very much alive. So if I want to go home and start reading about this, give me the best paper to start with. Who would you suggest?
So I'm happy to email all
Okay.
of them, but the people would be, there's a video if you want to look at the technique.
Just
reading.
overview of velocity recovery, the whole physiology of it.
The Zed Gragan, so him and Hugh Bostock did it initially, and Veronica Tan. So she's done all of the studies on the channelopathy patients.
Okay.
But I can email you a list of...
I'll pass you a card. Thank
Yeah.
you.
If you would like to know more about periodic paralysis, visit periodicparalysis.org. And if you enjoyed this video and want more, hit that like button, subscribe to our channel, and hit that bell so you don't miss any future videos. It really does help spread the word. You can view other videos about periodic paralysis by clicking the thumbnails to the right. If you have questions, just leave a comment below or reach out to us on social media. We'd love to hear from you.